Here we go. So as a quick reminder, on yesterday, what we had done was we had looked at how we turn DNA into RNA, which turns into amino acids, which turns into proteins, etc. The big takeaway here was learning how to properly decode all of this stuff here. I'm pretty confident you guys are able to do that again now. I won't kind of go over that again. I'm confident you guys will be able to do that. If you don't, please let me know, because this is like the culmination of 2 weeks of stuff here. So we're going to finish off this topic today. Here's my stuff. Yeah, so we're going to finish this off today, and we're going to look at gene regulation and mutation as well. So this is 12.4. Okay. So we are going to look at, again, how we regulate genes and what that means, but also we're going to look at how you mutate genes, which I think you guys have a fair idea about, like, well, what does it mean to mutate a gene? So we're going to start out here with what the heck is gene regulation? So gene regulation is when we have an organism, a cell, an animal, a pine cone, a plant. It can control which genes are transcribed depending on the environment. It can control which genes are transcribed depending on the environment. Think of a bunny in winter. What happens to it? Its fur coat changes. It is in response to the environment. Think about a pine cone. It will sit there dormant in a dry year. In a really wet year, it will then open up, for example. So gene regulation is where it looks at the environment and then it says which ones it wants to go forward. How it does that is through something called an operon. So this contains the proteins needed for a certain, we call a metabolic pathway. Basically, this is the fancy word for it. It's an off and on switch. It has all four of these things, but when you think operon, think on or off. It's waiting for the right environmental circumstances to turn on or off. It has other things, of course. It has a promoter. This is where that polymerase binds to. It has a regulatory gene. It has a gene coding for proteins, but at its core, you just know it needs to turn it on and off. Oops, sorry, I got to do attendance. I'm going to get yelled at. Okay, so there are two different kinds of operons. There are two of these. The first one is called a TRP operon. And what I want you to think about this is, I want you to think about this like a water dam. So a TRP operon is like a water dam. Basically, this is usually off. A water dam is usually mostly closed, right? Let the water build up behind it. Occasionally, though, we need to open that dam. We need to turn this operon on. So a TRP operon is usually, it's called a repressible operon, AKA it's usually off. So a TRP operon is usually off. A dam is usually closed. You will occasionally open it up. You will occasionally turn this on, but its normal state is closed. Normal state is off. And so again, this is normally off. Now I want to make sense of this picture down here. Where's my pen? So this picture down here, I will zoom up, zoom into this a little bit in a second. But all you really need to know is it is normally in the off state. This is the state on the right where it normally is. And you can see I have something here called a repressor. This repressor is usually in the way. The dam's gates are usually closed. When I remove that repressor, that little red part there, that's when, oh, I'm good to go now. I can go, it can move. Water will flow, right? I've opened the dam gate a little bit. So that's why I like thinking about this like a dam. So again, TRP, usually off. The other one is called an LA key, a lack operand. You don't need to know the first part here, but a lack operon is, it's called an inducible operon. I'll make sense of this. This is very scientific. It's an inducible operon because an inducer binds to the repressor and inactivates it, allowing transcription to be turned on. AKA, this is can be either on or off, but it will bind to a repressor and inactivates it. This is, what's a good example of this? This is like traffic over a railroad. The only thing is that there's those red lights that tell you to stop. So you can think about this as like crossing train tracks, I guess. So normally traffic's free to go, right? Normally cars go across it regularly. When those red lights come on though, you got to stop. So again, TRP operon is like a dam. It's normally closed, but you can open it. You remove that blockway, it opens. This is like train tracks. Normally traffic flows across it totally fine until you have those lights turned on. Let's see. Do we need much of this? Nope. Okay, Hox genes. This is going to make a lot more sense once you guys read the textbook. You will see this in your book. Read this. It will make so much more sense than I'm able to explain it. But think about like a fly with this. Basically, gene regulation, crucial for development and cell differentiation, yada, yada, yada. The big thing here is that we have groups of genes, and these control differentiation. And how this works is something called a Hox gene. So a Hox gene is a group of genes. These will transcribe at certain times on the genome, and they'll control what body part will develop at a given body location. So these are timed ones. They're on a timer. Not all of this is going to develop all at once. It might grow a certain part first. It might grow this, the green part first, then the blue part, etc. Again, your textbook does a really good job of explaining what this part is specifically. But the big part here is a Hox gene are timed genes. They are transcribed at specific times, dependent on where they are in this genome. Again, as it gets read. And they specifically will focus what body part gets developed. Okay. So I'm just going to take a step back here. Again, when I'm looking at this stuff, okay, it seems simple enough, right? I've blocked the hole so stuff can't get through, right? How does it actually work though? How does it actually stop it from reading this RNA and DNA? It's done through something called RNA interference. We call this RNAi. So this stops this messenger RNA from translating its message. Because you remember that central dogma, you go DNA to transcribe into mRNA to translate into protein. So this stops the protein generation. So it stops protein. How does this work? So these RNAis, they will bind to the mRNA and they'll stop it. So that's this part right down here. So there's nothing else for regulation. The next thing, of course, is mutation. So we already have a pretty good idea as to what can cause mutations. It reads it wrong, it inputs the wrong thing. There's an environmental factor, etc. There's a couple different kinds of mutation. I'm pretty sure you guys know what a mutation is. It's a change in your DNA, right? There are a couple kinds. We have point. This is when we just change a single base pair, a single pair. So maybe instead of an A and a T, maybe now it's, I don't know, it's a T and an A or a C and a G or a whatever. So I'm just changing one thing. And that can be done two different ways. We have missense substitutions. Your DNA goofed up. Your spell check didn't work. Or a nonsense mutation. Oops, I accidentally put that stop codon early. So missense, you give the wrong protein. Nonsense, you've given that wrong, you've put the stop codon in. Remember, this is going through the ribosome, right? This is creating those proteins. So a point mutation is changing one of those pairs, one of those proteins, and that's it. The other one is an insertion or deletion. This is when we add or lose some or many nucleotides. So again, so we're just going to be focusing on mutations and what some of these things mean. So one of the things that can cause a mutation is when those small changes in DNA codes can create genetic disorders. Sometimes just as simple as one single protein. A single protein can cause these disorders, or it can cause an allergy, for example. A lot of allergies are caused by a very small genetic mutation. So when we change one of these acids, this changes the entire sequence of the protein. And this can affect the stability of the entire thing. Now, of course, what causes mutations? We've looked at these before. Sometimes it just happens. Your polymerase, and this is very rare, it does mess up. Keep in mind, throughout your lifetime, you will be copying probably tens of trillions of genes throughout your lifetime. It's gonna mess up sometimes. Otherwise, chemicals and radiation, right? Carcinogens. Those can cause, they're called mutagens. These can damage the DNA. not so important to know like how they work. You just kind of need to know that they do that, right? The big thing here is just understanding what are some things that cause mutations. Okay, and so last thing here is the difference between a body cell and a sex cell mutation. What does that mean? So you don't pass along every mutation. If, for example, let's say all of a sudden something went wrong and it starts spitting out a different colored hair for me. I was born, I don't know, I got exposed to some chemical and boom, now my hair started changing color. That does not necessarily mean that I'm going to pass that on to my kids. That affected the hair follicles. That did not affect the gene that controls what color my hair is, for example. So if it's, we call it a somatic cell mutation, those are not passed to the next generation. A body cell is not passed. But if something happens in our sex cells, that can get passed on and will be present in every cell of the offspring. Okay. So that's chapter 12. We will end up looking at human genetics next week a little bit. But otherwise, we'll be kind of done with genes, probably by Wednesday. Okay, so yeah, I'll give you a minute for these 4 questions here. Not a whole lot to write. You're only going to need like 2 lines for each, and even that's kind of like a lot. So this won't take too long. So what is gene regulation? Gene regulation is the ability of an organism to control what genes are transcribed in response to the environment. What's the 2 types of mutagens? Radiation and chemicals are the primary ones. Again, your body can get it wrong, but it's much less common. Sorry, much less common for that to happen. It's rare that your body will get it wrong naturally. Next is we're looking at something called a point mutation. If you remember, this is where you got like where we mess up the order of the protein. In specifically in a hemoglobin gene in your blood cells, this will cause the protein to fold abnormally. The protein is it can reduce its stability, basically. And then the next one is, why are most mutations in eukaryotic cells recessive? Most are recessive. In the prokaryotics, those are controlled by those operons, those on-off switches. But in eukaryotics, we have a lot of those regulatory genes. Okay, so that's it for unit 12. Oh, come on. So next week we're gonna go really quick next week. In fact, I'll probably print off most of the notes, to be honest with you. I'll do something this weekend to figure that out, but we got 3 sections to go through still. So we're gonna move pretty quickly. We'll probably have 1 day for review. I'm gonna try and give you guys 2 as much as I can here. So for the rest of the day, there's 3 things. One, and I know sometimes, it's not, we go through the textbook and it's like, okay, I didn't really gain much from this. 12.4 is a really good one to read. You want to go in there and read it, especially that fly part. So one is you're going to make sure you finish those study guides. You can use that was what was handed out earlier. You want to go through and read 12.4. So those are the first 2 things. Read it, do the study guide, hand that in. The third thing is I've just opened up that quiz here as well. So that's what today is. And then if you haven't yet, please make sure you submit that codon assignment, which is this thing here. It's just translating all this from DNA to the 2 kinds of RNAs, the codon, the anticodon, and what protein that spits out. I'll let you guys get started with that. Turn that